So I believe that we can start. Welcome to the Hellenic Pasteur Institute. As you know, today it is the major event that the Hellenic Pasteur Institute celebrates the bicentenary of Louis Pasteur's birth. You know that here in Athens we have done a few activities. Institut Français has done it. We also did another informal exhibition a few months ago. But today it is the main event. Before I start, I'd just like to salute our distinguished colleagues, the president of the Hellenic Biochemical Society, several professors, several friends, several colleagues, people who have been associated with Pasteur directly or indirectly throughout the years. So I would like to call Professor Grice from the Pasteur Network, just to say a few words about Louis Pasteur, the Pasteur Network, and whatever you like. And thank you very much for being with us on this occasion.
Thank you. I want to first thank Dr. Statis for his invitation, and especially for allowing me to come visit and meet all of you here, for those of you that I've met. So my name is Rebecca. You don't need to call me doctor, professor, any of that stuff. And I started a new chapter at Pasteur. And Louis Pasteur, in his great foresight, said many wise things, which are sometimes attributed to him. We're not entirely sure he said them, but one of the things that he said was, science has no country. And in order to fulfill his original legacy, which was to ensure that scientific knowledge, which was then translated into benefits for human health, is to make sure that all of the work that he did in establishing different networks and different institutes around the world that he did, as well as his disciples, comes to fruition and is modernized today. So in 2021, there was a change in the Pastoral Network, and that change was meant to ensure that all 32 of the institutes, which doesn't include Pasteur in Paris, that would be 33, are participating and are appropriately represented as a part of the Pastoral Network, so that we can harness all of this work, which is across all continents and all regions, to address global health challenges. And IPH here plays a critical role in that. Greece is at the intersection of East and West. It plays a critical role in terms of science, which we hope to ensure is elevated and promoting through excellence in all of the work that's done here. And that there's a bigger contribution for IPH in terms also of helping its colleagues in the network, and it plays an even bigger role in the landscape, not only in Greece, but also across Europe and ideally in the world. And it's a pleasure for me to be here. I hope and wish that all of you participate. You know that we appreciate and think of you all of the time, believe it or not. And you're more than welcome to address anything to me or to anyone else at the Pasteur Network in terms of other opportunities that you might have, in terms of improving, addressing, and ensuring that your own individual careers and the progress of IPH meets all of your expectations. So thank you so much for having me here today.
Thank you very much, Rebecca. We also have Philippe Grier from the Institut Français here in Athens. So Philippe, please come just to say hello on behalf of the Institut Français. Yeah, just hello. In as many words as you wish.
Okay, hello everybody. It's a pleasure for me to be here tonight and to represent the French Embassy. And I am myself a professor. I was a professor at the University of Toulouse some years ago. Now I work at the French Embassy, and I'm very happy to be here tonight to hear a nice conference from Nobel Prize. Thank you very much.
Thank you very much, Philippe. Well, I don't think that if a person has so many achievements like Aaron needs a real introduction. I would just go through very, very briefly. He was born in the so-called British Protectorate of Palestine at the time, because his parents have moved from Poland to the state of Israel. It was just part of Britain at the time. He did his NMD, a medical doctor, in Jerusalem, in Hebrew University. Then he spent several years doing his military service. I mean, you know, he comes from a country with very sensitive neighborhood. And then he continued doing his PhD in Technion, correct? Afterwards. And then he left Israel, going to the States, to the Fokker concepts, even by extremely important people at the time. I just like to skip their names, that they were thinking that proteins are static. Once they're made, they're there forever. So Aaron, he did this clever approach just to go through and say, no, this cannot be the case. I mean, nowadays it is obvious to all of us that proteins cannot live forever. And then they move just to study what is happening right after a protein is being made and what is the turnover. So in a way, he opened a totally new field of selective proteolysis, if I may say. And here I have to say something for the young people, but also the senior ones, that his major discovery was published in BBRC. And I say that because I'm embarrassed to hearing here and outside Pasteur that unless you publish gold open access, you will not get citation and your work is not going to be recognized. And just to use Aaron's his own word, it is not where you publish, it is what you publish. You have said that, I remember. So this is exactly what Aaron has done, and you will see how a totally new field has been developed, which to my understanding, it is perhaps more important because at least human beings were on post-mitotic cells, so protein regulation by degradation, it is equally important. I mean, Aaron, of course, has received many awards. I mean, there is no need just to go through that. I will spend another 10 minutes going through all that. He's a very good personal friend. He's a very good friend of Greece. He has come to Athens on several occasions, even to schools, or we had a public lecture to Megaron Plus, to universities. And of course, he's a friend of Spetses, the famous island where the schools on molecular biology take place since 1996. And actually, we've been even together on peculiar occasions, like on September 11th. You remember, Aaron, we were in the Anargiri beach together. And that was a time almost before internet, so for several hours we were in isolated beach and we didn't know what is happening. So, Aaron, thank you very much for coming here. I'm sure that everybody is going to enjoy your lecture, and it's our privilege having you at the Hellenic Pasteur Institute. So please come.
Okay, good evening, everybody. Thank you, Statis, for the invitation. And it's a great honor for me to be here today because of this special event of the second centennial for the birth of one of the greatest scientists that ever walked on the face of Earth. We need to understand that 200 years ago, there was no science the way we know it these days. There was no communication, no instruments, no microscope, almost nothing. And people were on their own, so there was not even a formal educational system. And so he was clearly a unique figure on this landscape, and his contributions are truly immense, truly immense. Today it became much more crowded, much more systemized. We are living in a much more ordered and organized systems, but this was the era of the real giants of science that in many ways has passed. I mean, we are working on corners, important corners. We are adding a lot to our knowledge, developing vaccines and so on and so forth. But at that time, I think you could count on one hand the number of people that were really prominent in science, and they did it on their own, no frame around. They had to build everything by themselves. So for me, these are my heroes in many ways. Among them are, of course, Newton and Darwin and, this type of people that, What I want to tell you today is the story, our own story. I will, because the audience is so broad, I will avoid going into technical details, but I'll tell you more about concepts. How we came, what did we think, and how it was discovered. At that time, we didn't know what will happen in the future. Of course, science is one of the most wonderful professions in which you are moving from corner to corner, and you never know what's, thank you very much, and you never know what's behind the next corner, and just time tells you, you know, what, time tells you what, you know, whether it's important, whether it's novel. There is always this joke about somebody who sends his paper for publication, and the editor comes back and says, your paper is both important and novel, however, the novel part is not important, and the important part is not novel. So, so you never know. You know, it's a joke, but it has a serious nucleus of truth in it. So you never know how things are going to evolve, and typically, it's the work of others that take whatever you discovered and take it further. And what happened to us, we were, as you'll see, we were interested we were driven by curiosity, we identified a small, opened, unanswered corner in science that we decided to study. And then we discovered something, and then people thought, wow, maybe such a system has also clinical implications, so they started to study diseases, so diseases were discovered, and then the pharma companies walked in, and now it's a big industry of drugs and affecting millions of people worldwide, and I'm going to tell you about the whole story from 30,000 feet height. So this is, how do I change here. So this is the beautiful city of Haifa where I was born and working, and you see the medical school. Here is the medical school, and this is our teaching hospital, and these are the beautiful Baha'i gardens. Yeah, yes, you can see it from here. This is forward. Or here. Okay, perfect. So let's go back. So this is the harbor of Haifa, and Greece is somewhere here to the west. Cyprus, and then up north is Greece. So we are about proteolysis. And in order to understand, proteolysis is a destructive process, as you know. We are destroying proteins. We are basically introducing a molecule of water in the peptide bond and split it. The two amino acids are being split, and proteolysis occurs in the body at several levels. In the stomach or in the gastrointestinal tract, we are digesting proteins that are coming from the diet, you know, meat, cheese, whatever. And the reason is simple. We cannot introduce into the body intact foreign proteins because we shall elicit an immune response. Now we are moving into the body because the gastrointestinal tract is basically extracorporeal. It's a tube that's crossing the body, but topologically it's outside of the body. Now we are moving into the body, and in the body, you know, we are still at the extracellular environment, but intracorporeal, and there are many proteolytic systems. One of them is, of course, the blood coagulation system. The blood coagulation system is a cascade of proteolytic events where you have an inactive protease that is being cleaved by an upper protease to a smaller fragment. The smaller fragment becomes active protease, and the cascade goes on all the way down until fibrinogen is converted into fibrin to make the blood clot. Then we are moving into the cell, and into the cell there are also proteolytic systems. One of them is, of course, the lysosome, on which I'm going to talk, and to tell you how we made a distinction between what we are looking for and the already known lysosome. Now, one common feature of all proteolytic systems is they are destructive. And in order to protect ourselves from destruction, we have to evolve some mechanism that will protect us from uncontrolled destruction. So in the gastrointestinal tract is the mucinous secretion that is lining the epithelium of the gastrointestinal tract. If the continuity of this mucinous layer is disrupted, we get a disease called ulcer. You know, and the ulcer can be in the stomach, it can be in the duodenum, in the small intestine, and even in the large intestine in rare cases. So this is a protection. Here, in the intracellular, in the intracorporeal system, the protective mechanism are tissue factors in the coagulation. So when we get wounded, we don't coagulate the blood all over the place, because if you coagulate the blood all over the case, It's fatal, it's a disaster. Even if we coagulate the blood in the coronary arteries, we get myocardial infarction, we get a heart attack. So we want to use blood coagulation only to protect from severe bleeding. So we are secreting tissue factors, so it's about localization, and these tissue factors are activating the coagulation system only in the place of the wound, not anywhere else. So this is another way. In the cell, when we are talking the lysosome is an organelle. I'll show you the lysosome. Here is the lysosome that was discovered by this wonderful scientist, Christian de Dive, a Belgian-American scientist. And the lysosome, here you can see the lysosome in magnification. This is the cell. You see the mitochondria here. And the lysosome is a membrane-surrounded organelle. So all the proteases are inside. They are inside, they are not outside. And the target proteins have to enter the lysosome in order to be degraded. So typically they are entering the lysosome either from the outside, being carried out by endosome, by series of vesicles that fuse to the membrane of the lysosome, or the lysosome pinches off by a process called microautophagy, droplets of the cytosol. You can see it here. This is the pinching. These droplets become vacuoles, and then the vacuoles open and pour their contents into the lysosome to be degraded. So the mechanism of protection is double. Once we have a membrane, so these proteases, these destructive sharks, are not floating freely in the cytosol, and two, the pH of the lysosome is low. It's 5, while the pH of the cytosol is around 7. So if the lysosome by any chance explodes, it's neutralized immediately. The pH is neutralized immediately because the lysosome has a very small volume, and the volume compared to the volume of the cytosol, and the proteases are all acting optimally at low pH. Once they are encounter the neutral pH, they are inactive. So you see, nature evolved all along the way protective mechanism. Now we are interested in what happens here in the cytosol. Now, why we were interested in that? Because if you look here, this droplet contains all the proteins that are in the cytosol. Like if I take now a nylon bag and I will collect air from this hall, from this lecture hall, there will be molecules that are coming from all over that were expired by all of you. Let it be nitrogen, let it be carbon dioxide, let it be oxygen, and I will not be able to make any separation. It will be a mixture. So a mixture of all the representative proteins that are in the cytosol are contained here. Once it's opened for degradation, they will all be degraded. We are interested in specific protein degradation. So a priori, we knew that there must be specific process. What is specific? Specific is that you have 1,000 molecules of the same species, and there are millions of molecules of the same species of each species. So let's say RNA polymerase, DNA polymerase, hexokinase, doesn't matter. They are not a single molecule in the cell. There are millions of them, tens of thousands, hundreds of thousands. Let's say that one of them get misfolded or denatured or mutated. We need to pick up this single one out of the whole crowd, degrade it, and keep all the 999 intact. This is a completely different ballgame. How the cell evolved, pair of spectacles that will be able to pick up one, and there is no way that this one will be in this vesicle, because this vesicle contains millions of molecules, to do it. So a priori, we knew that there must be a specific mechanism for their degradation that is very distinct from all known other proteolytic mechanisms. You know, trypsin, chymotrypsin, all these enzymes that degrade proteins are just cleaving enzymes. We knew that we have to look for something else. One feature of this something else is energy. Typically, energy is used in order to keep cascades of ions and so on and so forth, but also when the cell have to exert specific processes, to exercise specific processes, it pays with one coin, energy. But proteolysis is not an energy-requiring process. It's the opposite. When we are eating proteins, we are eating them in order to gain energy. Remember, if we eat sugar, we get 4 kilocalories per gram. If we are eating proteins, we get 4 kilocalories per gram. If we are eating lipids, we get even 9 kilocalories per gram. So proteins are source of energy. But here in the cell, it's the opposite. We knew that we have to invest energy. Why? Because there is a specific process there. So a priori, we knew that we are looking for something that is completely opposite, not only conceptually, but also thermodynamically. So these were the guidelines that guided us when we started our research. Now, I was a graduate student at the time, which tell you that graduate students still have hope, so don't give up. So you can make up it all the way to Stockholm at the very end. And with Avram Hershko, a very talented biochemist that just returned himself, he was a young scientist at the time, he just came back himself from a postdoctoral fellowship in the United States, and I was starting my internship, my residency in surgery. Actually, I graduated medical school. It was exactly after the war, this 1973 war. There was a big war in the Middle East at that time, and I was a military physician at the time, and I was under the impact of the war, and I decided to become a surgeon, you know, to see how things are really happening in the operating theater. But then I thought that it still might not be for me, because I wanted to understand the source of diseases, why diseases start, rather than how they end. Because when you see a patient, it's in most cases too late. Most of the patients that you see, it's too late. When a patient comes with a heart attack, he already has occluded coronary artery in a process that lasted before the myocardial infarction occurred for decades. If you are seeing a patient with Parkinson's disease, decades have elapsed since the first pathological event. Already 80% of the substantia nigra par compacta has gone already. When you see a patient that complains about an abdominal pain and you discover pancreatic cancer, it's too late. The tumor has been there developing for decades, especially for pancreatic cancer, but even other cancers. It takes a long time. And I was very naive, thinking that, okay, I want to understand how diseases start from the very beginning. In retrospect, I thought that it was a childish type of a thinking, because at the end, once you are coming to the lab, you are concentrating on a very narrow corner of science. You are not studying big diseases. You are not discovering drugs. But I was lucky at the end, as you'll see, to see the process evolving not from our side, but from the side of the scientific community. So this is the lysosome. And what really drove us was this, part of it was this table. So what is this table? It's the half-life time of different proteins. Typically measuring what we are doing, we are measuring kinetics. So every protein has its own half-life time. What is half-life time? All proteins are degraded. I'll talk about quantity in a minute. And each of them has a half-life time. What is the half-life time? It's the time that is required to 100 molecules to go down to 50. So how long it takes to halve the amount of the protein. Of course, typically proteins don't go to half, but they exchange. So the 50 molecules that are degraded are replaced by new ones. But nevertheless, if I label 100 molecules, I can measure the decay of the label. So this is one kind of a measurement of protein dynamics. And you see that protein dynamics is very variable. You know, this protein has a half-life time of 12 minutes, while this protein has a half-life time of 15 hours. And again, all these proteins are swimming in the cell in the same very aquarium. So how is it that each of them has a different half-life time? And furthermore, as I told you, this molecule is represented in the cell by millions of different copies of copies. So again, if one molecule of these proteins or of any protein get mutated, misfolded, denatured, it's being picked up, and the half-life time is even accelerated. It's being removed immediately. Why? Because we don't want to keep inactive molecules in the body, and we'll talk about it once we talk about cancer, neurodegeneration. These are accumulation of untoward protein that were not degraded properly. So this was one of the guidelines. And actually, I would say that the father of our hypothesis, we were under very tough competition at the time, was a wonderful scientist by the name of Brian Poole. He worked at the Rockefeller Institute, and he did a very simple experiment. I think that this is one of the most elegant. You know, once you are looking for elegant experiment, look for the simplest one. Don't go to the more complicated one. This was one of the most elegant experiments I've ever encountered in my career. What he did, he took two dishes. He took two dishes of cells. One dish he labeled with tritiated leucine. So leucine is an amino acid that is labeled with one isotope, and the other one he treated with leucine that is labeled with C14. It's another isotope. So the cells don't care about the isotope. They don't know what we are doing to them. It's the same very amino acid, and it goes all the way into proteins. But energetically, tritium emits much lower energy than C14, so you can, in your spectrometer, you can say which is which. You can say, wow, this is C14, and wow, this is tritium. The cell doesn't know about it. Only we know about it because we developed instruments to read windows of energy. Then he took the C14 labeled cells. After they were labeled, he washed them, and then he destroyed them. He ground them in an homogenizer. And he took this debris, and he put it on top of the intact cells that were labeled with tritium. So now, the intact cells that were labeled with tritium are degrading two types of proteins. They are degrading their own proteins, because these are intact cells, but they're also degrading the C14 labeled debris that they are taken from the outside by pinocytosis and endocytosis. So in this way, he was able to measure in one type of cell the degradation of two distinct populations of proteins, internal and external, endogenous and exogenous, and to follow the kinetics by using a spectrometer that can read different windows of energy. And he used, on top of it, then he divided the culture into 4 dishes, and he used control, of course, without nothing. He put nothing, and then he used chloroquine. Now, chloroquine is an antimalarial drug, and it neutralizes the lysosome, the pH in the lysosome, so there is no degradation of the lysosome. So he either used cells that are in which the lysosome is active and cells in which the lysosome is inactive. And what he found was amazing. He found that the endogenous proteins, once he adds chloroquine, almost nothing happens, little inhibition. Here even less, 4%. But the exogenous protein, the one that are labeled with C14, the degradation was vastly inhibited by chloroquine. So he concluded that the exogenous proteins are degraded in the lysosome because a lysosomotropic agent, an inhibitor of the lysosome, inhibit them, while those endogenous proteins, these are the subject of our study, are degraded by some other system. And Brian, I came to know him, unlike many scientists, he was also a poet. He composed songs, and it's very rare for scientists to be artists or to be poetic in their spirit. And he formulated it in an unbelievable way. And that's what he wrote. Without going into the experiment which I explained, he said the exogenous proteins, those that are coming from the outside, will be broken down in the lysosome. Why? Because we can inhibit their degradation by chloroquine. While the endogenous proteins will be degraded, will be broken down wherever it is that endogenous proteins are broken down, somewhere, wherever it is. He didn't know what it is, so he called the elusive system wherever it is. It is that endogenous proteins are broken down during protein turnover. And he was looking for this wherever it is, but he died, unfortunately, tragically, from complications of diabetes. And we were on the way. We competed without knowing even that we are competed, and we started our search, as I said, under the guidance of Avram Hershko. And because we are both physicians, we went to a model, a very simple model, which is called the reticulocyte. Now, the reticulocyte is a young red blood cell that still maturing to become the mature red blood cell, and the maturation involves degradation. It ejects out some of the systems, like the nucleus and the mitochondria and so on, but it degrades other systems, like the replicating enzyme, the cell cycle enzymes, and at the end, you are getting a very primitive cell. The red blood cell is kind of a very primitive cell, very important, but nevertheless primitive. It has mostly hemoglobin, of course, the red blood a protein that carries oxygen and carbon dioxide. It has the glycolytic system in order to produce energy, glutathione, cytoskeleton, very, very few functions are being left. So we said the reticulocyte will be a very good system for us, and we knew already that the reticulocyte doesn't have lysosome. Remember, this was 76, and in 76 there was no siRNA, no shRNA, no molecular biology, no nothing. There was only nature that provided you with systems to work on, and we used the power of nature in order to go after the system. So we took the reticulocytes, and we knew that there is no background of the lysosome, and we looked for the system. And as Statis mentioned, we published our first paper in BBRC. And this paper has some nice features in it. First of all, at that time, you know, it's a low impact factor paper, but at that time we didn't know that it's a low impact because there was no impact. It's because there were no computers, so people couldn't calculate any impact. It was a journal. We knew that it's not that famous, but, you know, people could not enumerate quantitatively the value of this guy. By the way, this is the paper that was cited by the Nobel Committee. So just to tell you, again, as Statis said, it's important what you publish rather than where you publish. But there was another issue that I want to emphasize about this paper, and which really tells you how we were enslaved along the years with bibliometry. So Avram, after I started my PhD, 7 months later, Avram left. He went to sabbatical in Philadelphia. He left me alone. So I was working in the lab, and then I sent him the results. And he wrote the paper, and he didn't tell me even. Avram is a cool guy. And he sent the paper directly for publication. He didn't even show me nothing. So the paper was sent and published, and he said, Aaron, I'm happy to. And at that time, again, there was no even fax. There was nothing. All the correspondence were with, you know, these blue envelopes that has red and blue colors on them. It's called paravion en français. It was all paravion, and it took like 2 weeks to go each direction. And 20 years elapsed. I'm coming to my last promotion to become a full professor in my institute. And there was a committee. I didn't know even that the committee is convening. And I, of course, subjected my papers and my research program and my research history. And one angry professor of pharmacology walks into my office and he said, hey, Aaron, the paper that you claim is your founding paper of the system that you discovered is not on your name. I said, what? So he showed me the paper. He pulled out this paper and he said, it's not on your name. Look in PubMed. At that time, there was already PubMed. It was the early 90s. So I looked in PubMed and it did. It was not there. I said, you know, these people in PubMed must be crazy. You know, the computer went wrong on my paper. And I looked and looked and looked. It took me 20 minutes until I discovered that Avram spelled my name with a mistake. So you see here, well, first of all, he put a H here. Aharon is the biblical name, how it's written. But in English, it's double A. It's A-A-R-O-N. There are two words in the English language that are spelled with double A. Aaron and aardvark. That's it, this animal. That's two. You go to the Oxford Dictionary, that's what you find. But then he missed a C here. It's Ciechanover. It's not Ciechanover. And of course, PubMed, you know, it's a PubMed. It's a software. It's the, you know, if you are missing a letter, it doesn't show up. So I went to this name, and indeed, this guy published one single paper, this paper. And this paper is cited several thousand times because it's the founding paper. And then he was convinced that it was a mistake. Avram really, you know, he even didn't know my family name. And he left and he sent the paper and so on. You know, it's kind of a joke, but it's really a sad joke because it tells you how unattentive we became to people. We don't ask them what they do. We want to know how many Nature papers they have, how many Cell papers they have. We don't care whether it's important, whether they tell a story, whether they don't tell a story. We measure them by numbers. Give me your number, give me your H factor, give me your citation, and I'll tell you who you are. If not, if you are not good, don't come and tell me any stories. Go to tell them to your mom. You know, I'm not the guy to hear them. So anyway, there was, well, it's stuck. We are stuck. We are stuck. Somebody can help me with this computer? Well, I can do it without, you know, but it will be a little bit of a challenge. Oh, here it is. Okay, so this is the most important experiment we have ever done, and the rest is history, because the rest was derived from it. So this is a table, a simple table, that tells you what we did. We took the reticulocytes, and we lysed them in water. We spun down all the membranes, all the dirty stuff, and we used the cytosol, just the upper fluid. And we saw indeed, and we used in this case tritiated globin. So this was our protein, the target substrate that should be degraded. And we saw indeed that the lysate of reticulocyte that has no lysosome at neutral pH, this experiment was done at a pH of 7-8, did degrade proteins independence of energy. So that's what we were looking. We were extremely happy with this experiment because we had at hand the system that we were looking at. We were looking at non-lysosomal, ATP-dependent proteolytic system. And we had it. Then we said, okay, we are biochemists. There is no molecular biology. The name of molecular biology was not even in the dictionary at that time. We said, okay, let's purify this elusive protease that is an ATP-dependent protease. So we took the whole lysate and we put it on a column. Doesn't matter, it was an anion exchange column, doesn't matter. And we fractionated it to protein that adsorbed to the column and had to be eluted with salt to protein that just fell through without any manipulation. So fraction ones are the fraction of protein that came through. It didn't attach to the column. Fraction 2 is the fraction that we eluted with high salt. And you see that none of them had any activity. Well, fraction 2 had little. But when we combined the two of them, we got the activity back. 1.5, 10, 1.6, 10.6. So this was the whole story. This was a paradigm change because we needed two factors in order to get a proteolytic activity. There is no known protease that needs two factors. Take trypsin, it will degrade proteins that have lysine and or arginine at their end terminus, at the end terminal part, end of the amino acid. Take chymotrypsin, it will degrade protein that have tyrosine or phenylalanine at the end. There is no precedent for that. So once you have no precedent, you have advantage and you have disadvantage. The advantage is that you can save your time and you don't have to go to the library because you will find nothing there. The disadvantage is that there is no knowledge to hold into. I mean, you are really in a new field and in a new area and you have to work it by yourself. So everything else was new. And another thing is that, you know, the reticulocyte, despite being primitive, still has several thousand proteins. You know, a normal cell in the body has about 25,000 proteins. So the reticulocyte has about 2,000 proteins. So it could be easily that the fraction 1 has 1,000 proteins, let's say just roughly, doesn't matter, and fraction 2 has another 1,000 proteins. So who told us that there is one protein, one active component here and one active component here? It could be that fraction 1 has 1A and 1B, and 1A has 1A prime and 1A2 prime, and it's endless. And the same for fraction 2. So nobody told us that our system composed of only 2 proteins. It could be 3, it could be 4, it could be 40, it could be 400. It could be anything. So we started to fractionate and fractionate and fractionate, and when I left the lab in 1981 to go to MIT for my postdoctoral fellowship, I left behind about 12 proteins, 12, that were all needed in order to degrade a single target substrate. But not only that I left 12 proteins, I knew what they do. I knew the sequence of events, that I knew which is acting first, which is acting the second and the third. It was a very cumbersome work of kinetics and lag period. I will not go into it. But this was the limit of biochemistry. And I think that Avram, after I left, discovered another few, but then we had to wait until 2000. And what happened in April of 2000? Nature published the first human genome. This was a big celebration. And then it was discovered that the system that we discovered contains not one protein, not 2 proteins, not 20, not 200, but almost 2,000 proteins. 2,000 proteins. This system that garbaging other proteins is containing. And how they discovered? By homology, by families. And I'll show you roughly the families because I don't want to go into details, but by structural and sequence families, and sequence of families. And it's amazing, this discovery, because it's not only garbaging, it's much more complicated than that. And the basic principles of the systems are the following. For those of you who are familiar with the system, just I'll give you the basic principle. So this is the target protein to be degraded. It's like, it's folded like a wool ball. In order to be degraded, something has to happen to it. And remember again that there is a shark in the system. Now we have a shark already. We have a system, a proteolytic system that floats freely. But the shark should not recognize any proteins, but only proteins that need to be degraded, that are destined for degradation. And in order for a protein to be destined for degradation, it has to be marked. So this is already the first unraveling of the hypothesis that we posed. In this case, it's double phosphorylation. Doesn't matter. I like this protein because I've been working on it for more than 20 years. It's I-kappa-B alpha. It's the inhibitor of NF-kappa-B, which is a transcription factor. That doesn't matter. Something has to label it for degradation. So the label here, in this case, it's 2 P's. It's phosphorylation. Once it's labeled, because it has to be degraded, because it's mutated or denatured, and there are other class of proteins that are degraded, which I'll talk about in a minute, it's being bound to an enzyme called E3. Now, E3 is a ubiquitin ligase. You'll understand in a minute where the name is derived from. And there are about, out of the 2,000 components of the ubiquitin system, almost 1,000 are E3s. Why? Because they need to recognize different proteins for degradation. One that have 2 P's and one that have 1 P, one that is folded like this and one that is misfolded like this, one is denatured like this and one that is denatured like that, and so on and so forth. It's a kind of a police. Think about the police in Athens. There are 100 thieves in Athens or in Tel Aviv, doesn't matter, and there are 10 policemen. And the policemen are, each of them is assigned to either a region in the city or type of a crime in the city, or doesn't matter. So this is exactly the same thing. You have less controllers, less policemen than criminals, but at the end you do need a police power. So you need a policing power in the cell that will look around for quality control problems and will grab the ones that are deviating from their natural structure. So what happens is the following: a small protein called ubiquitin, it's a highly evolutionary conserved protein, is being activated by an enzyme. There is one single enzyme in the database, or 2. It's still debated. By ATP, we identified the molecular mechanism here. Then it jumps to another protein called E2. There are about 50 E2s. You'll see later on the general structure of the system. And from E2, it's going to the target substance. And it doesn't go once, it goes several times. So the first ubiquitin attached to the target, the second ubiquitin to the first, the third to the second, the fourth to the third, and so on. It builds what we call a polyubiquitin chain. Poly, numerous, ubiquitin chain. This polyubiquitin chain is a molecular glue. It's a matchmaker. It's a matchmaker that binds the target protein to the scissors. Only now comes the shark. So everything happens above the shark. But in order for the shark to degrade a protein, it needs the protein to be marked twice. Once by the P's in order to be bound to the system, and then by the molecular glue, which is called ubiquitin. The molecular glue binds the protein, it brings it together to the appropriate vicinity, and then there is another activity of ATPases, so there is energy also here. It opens it up, and then it's being injected, and this is like the blender here. There is a blender here that rotates. This is the protease, actually. These are the proteolytic site, and it degrades it into peptides. It doesn't degrade it to amino acids. This is one of the major functions of the system. These peptides are being served by the immune system. We are all immunized, I hope, for COVID-19. It's the ubiquitin system. What happens is we are injecting messenger RNA that encodes a large piece of the spike protein of the virus. This messenger RNA is being uptaken by APCs, by antigen-presenting cells, and the antigen-presenting cells are using the ubiquitin system in order to cut the protein that is being translated according to the information, and these little peptides are then presented to the immune system. And only then it's being degraded to amino acids. So it's important that the protein will never be degraded to amino acid, because this is the only way, or one of the major ways, that the immune system can make a distinction between a self protein and a non-self protein. The self protein, the self peptide, nothing happens. They recognize them as friends, and nothing happens. If it's a foreign protein, any viral protein, let it be flu, let it be polio, let it be any bacterial protein, salmonella, that is being ingested by the cells, doesn't matter. The foreign protein will be generating peptides. The peptides will be recognized by the immune system, and the immune system will elicit an immune response. So one of the major functions of the ubiquitin system is to present antigens to the immune system, and besides many other functions. So meanwhile, the system became much more wide, and ubiquitin has cousins, and one of the cousins is Sumo. Therefore, I showed you this the Sumo warriors. Sumo is small ubiquitin modifier. It's just an acronym. And you see it's very similar. Ubiquitin has this structure. Sumo is a little bit larger, and there is a whole family of ubiquitin-like proteins that are serving other purposes. And gradually, it became clear that the ubiquitin system serves many other functions beyond just proteolysis. But when I think about it teleologically, I think that we came to the ubiquitin system from asking a biological question. We came from the bottom. We didn't know. People ask me, why did you work on the ubiquitin system? I said, I never worked on the ubiquitin system. I worked on a biological question. And ubiquitin popped up as a protein that was identified. Because there is no, this is also showing you the power of biochemistry, because there is no way the genetics analysis would have discovered this system. Because in genetic analysis, you could discover that if you are deleting one protein in the chain or you're mutating it, you are stopping the whole chain. You can stop protein degradation. But where this protein is along the cascade, you never know, because there are hundreds of them along the cascade. So you need really to take the system and to reconstruct it biochemically in order to get to the function. It's one of the classical examples that only biochemistry, even with nowadays technologies, can do it. Of course, we are using now much more modern technologies and so on and so forth, but in order to discover a multi-cascade, a multi-component cascade of events that that B follows A and C follows B and so on and so forth, you need really to take it apart and then put it back and then understand how it works. So biochemistry still has a justification of existence. And then there is a ubiquitin code. I will not go into all the chains. It became much more complex than we thought. And the architecture of the system is pyramidal. You have one E1. It's an activating enzyme. You have about 20 E2s. And then you have 1,000, close to 1,000 ubiquitin ligases. Why they are called ubiquitin ligases? Because they ligate ubiquitin to the target. And then you have thousands and thousands of target subsets that are being degraded. Now, when I talked about degradation, I reasoned it by damage to the protein. I reasoned it by mutation, by denaturation, by misfolding, and so on. This is only half of the story. The other half of the story is that we need to degrade normal proteins that completed their function. Think about cyclin. Think about cell cycle. It's driven by cyclins, but then it's being stopped by cyclin-dependent kinase inhibitors, and so on. So there are cyclin-dependent kinases, and cyclin-dependent kinase inhibitors. So nature, in order to assure directionality of, there are many ways to assure directionality of processes, like DNA replication, and so on and so forth. But the best way is destruction. Because if you make, you can do it by phosphorylation, dephosphorylation. Nature has found numerous ways in order to control processes. But degradation is unidirectional. Once you cut a protein into two, it's gone. You cannot glue it back. You have to go all the way back to amino acids and to the synthetic machinery. So critical processes are controlled by degradation. One of them is cell cycle. Actually, this was discovered and prophesied by the people who got the Nobel Prize 2 years before we got it, by Tim Hunt and others. And Tim prophesied that cyclins must be degraded in order to allow progression of the cells from along the cell cycle, from G1 to S, from S to G2, and to M, and so on and so forth. So he knew that there must be a mechanism. He just didn't know which one. Well, once he got the Nobel Prize, he already knew, because it was only 2 years apart. But he knew, he prophesied already in the early 90s, when they discovered the system, or the late 80s, that there must be a specific proteolytic system. So once these proteins are ubiquitinated, they are being shredded into the blender and degraded. So the shredder is a very complex enzyme, but nevertheless the specificity which we were looking is determined by two steps upstream to the shredder. The shredder has some specificity. I will not go into it. There are whole studies that are being run around the 26S proteasome, but nevertheless, the main burden of recognition of the target substrate is being carried by upstream 2 steps. Identification of the substrate as being a target for degradation, binding it to the system, and then marking it by ubiquitin for degradation. And the Nobel Committee called ubiquitin the kiss of death. So ubiquitin kisses the protein to its final destination. So now let's jump to medicine. Now, if you think of medicine, medicine is a profession of numbers. You know, think of blood pressure, think of glucose, think of sight, think of hearing, think of anything. It's all numbers. If you take a textbook of medicine, Cecil, Harrison, doesn't matter, Nelson, you know, the last 100 pages are all numbers. You know, what is the normal range and what is the deviation? And deviation is always a disease, or in many cases. So proteins are the same. Think of hemoglobin. You know, each of us should have hemoglobin at the range of, I would say, 12 to 16 grams per deciliter, right? That's the number. If it's going up, it's too bad. It's called the Guglielmo syndrome, which is a precursor of cancer. If it's low, it's an anemia. It can be due to bleeding from the gastrointestinal tract, one of the first signs of colorectal carcinoma. It can be lack of iron. It can be lack of vitamin B12. It can be anything. So numbers are very important in medicine. Blood pressure, glucose level, and so on and so forth. So proteins are either in the normal level, or if the ubiquitin system in the cell is hyperactive, it will go beyond the level, below the level, or if the system doesn't work properly, it will go above the level. So these are the pathologies, and that's clear. And the pathologies have affected, now we know, any of the reasons that are the reasons of degradation. Let it be quality control, which, misfolding, mutations, and so on, control of processes like cell cycle, differentiation and morphogenesis. You know, cancer is a disease of dedifferentiation. The tissue goes back to an embryonic stage, rapid division, uncontrolled division, and so on and so forth. It loses the tissue characteristics. That's what happens. And here you see from the cover page of Science, you see immunohistochemical staining for ubiquitin. You see this little brown flame. These are ubiquitinated proteins that are precipitating in the brain in juvenile onset Alzheimer's disease. Alzheimer's disease is a whole syndrome. It affects typically more advanced age people, but it also affects young people, and this is juvenile Alzheimer's. And you see that there is some constipation. So proteins are being ubiquitinated, but somehow they cannot be degraded. The proteasome somehow is constipated and cannot degrade them. Now let's go to cancer. Cancer is the disease that has been studied, I think, the most of all diseases. A lot of effort is being put into cancer because it's #1 killer. I don't know about Greece. In Israel, it's #1 killer. The statistics was just, it's kind of back-to-back with heart diseases, with vascular diseases. 1 year with the hand of, one disease is the upper hand, 1 year it's the other disease is on the upper hand, and so on and so forth. So if a tumor suppressor like p53 is excessively degraded, we get cancer because we lose protection of the genome guardian. If an oncogenic or growth-promoting protein is under-degraded, we get cancer. So you see that any perturbance in the rate of degradation of critical proteins will lead to cancer. Now let's go now to specific examples. Think about human papillomavirus-induced uterine cervical carcinoma, one of the most common cancers in women of the cervix of the uterus. It's being caused by a virus called the human papillomavirus, the HPV. Now there is an excellent immunization, and mothers all over the world are encouraged to immunize the daughters against it so they will not catch the virus. And what happens, what is the mechanism of action of the virus? It encodes an early gene. It has an early genes and late genes. One of the early genes is E6. It's called E6. Now E6 has the property, think how sophisticated viruses are. E6 meets p53. It combines to p53. It makes a heterodimer with p53, which is a good protein. It's a tumor suppressor. Now the ubiquitin system looks at it and it says, hey, something is strange here. And it degrades p53. It thinks You know, it's a misleader, actually, the E6. It thinks that the P53 is an abnormal protein that should be flushed into the sink, and it degrades it. It's like if you want to break into a bank and to rob it, you don't go to the safe. You first go to the alarm system. And P53 is the alarm system, so you first turn off the alarm system. Because P53 senses damages in the DNA. It's a sensor. Because the virus is a small DNA virus that integrates into the genome, into the cellular genome DNA. So in order to integrate, it has to make a cut. And once it makes a cut, it puts its two ends into the cut and integrates. This cut normally is being sensed by P53. But if there is no P53, nobody can sense the cut. So the alarm system is turned off, and P53 goes on and induces the malignant process. This is one of the malignant mechanisms of P53. And here you get a cancer. So this is, again, it's misleading the ubiquitin system to think, deceiving the ubiquitin system to think that P53 is an abnormal protein because another protein is linked to it and it degrades the whole thing. Let's look at the other side. The other side, we can take, for example, the HER2 or the EGF receptor in breast cancer. You know, the drug Herceptin that is being used to treat her. So what happens normally, once the EGF receptor binds to once the EGF ligand, binds to the receptor, it sends a signal. That's the normal activity. The signal leads to signal, of course, that we need the signal, but it's simultaneously, it also leads to ubiquitination of the tail of the receptor. And the receptor is being endocytosed, and the ubiquitination now leads to lysosomal degradation. In this case, it's not the proteasome, because the ubiquitin system, we learned, can send proteins to different proteolytic systems and to different destinations. Now, what happens in breast cancer, in mutated HER2 or mutated EGF receptor? The receptor is truncated. It cannot bind the ligand, but it signals non-stop. It's not degraded because there is no signal for ubiquitination, there is no ligand, but it signals non-stop to the cell, and the constant uncontrolled signaling is the leading event in the malignant transformation. So it's not degraded by the ubiquitin system because there is no normal ligand that binds to the binding site of the receptor because it's truncated. And what Herceptin is doing, it's actually clusters the receptor, and the clustering leads to ubiquitination. That's the mechanism of action of this antibody, that it clusters the receptor on the cell surface. So here you see that a growth-promoting protein, once mutated and not degraded, will lead to cancer. So you see, and there are many examples on each side of this equation. Accordingly, drugs were developed, and one of the drugs that were developed is Velcade. I'll talk about it, and the drugs are simple initially. The initial drugs are inhibitors of the proteolytic sites. Here you can see this cage, the blender. You can see in red are the active sites, and the drugs are inhibitors of the active sites. And one of them is the boronic acid derivative. It's called bortezomib. This is one of the old ones. And it's being used mostly for one type of cancer, and this type of cancer is called multiple myeloma. Multiple myeloma is a B-cell leukemia. It's a very common cancer. You know, it's about 3% of all malignancies and about 20% of all hematological malignancies. So it's quite common disease. I will not tell you how it was discovered, but I'll tell you about the mechanism. And what happens is it's a B-cell leukemia. So the B cells are secreting high levels of immunoglobulins. So you see the patient. In this case, it's a monoclonal expansion of a B cell. You see the galloping level of the immunoglobulin. Here, chemotherapy started. There is a little bit of relief, but then the disease relapses. And here you start treatment with Velcade, with the drug, with the proteasome inhibitor, and you go down to a complete response. So the patient responds very favorably. And you can see here the bone marrow. Here you can see the bone marrow of the patient. You can see that it's very homogeneous. This is a malignant bone marrow, and the tumor burden is 41%. Following treatment, it goes down to less than 1%. And this is almost a normal bone marrow. And this is a single drug therapy because this was taken from the clinical trials. We never treat cancers or leukemias with single drugs. It's always a combination. And the disease really changed its clinical landscape. I remember as a medical student, people died from the disease within a year in agony. What happens, the whole bone marrow is being occupied by the B cells, so it pushes all the other elements of the bone marrow, The patients are suffering from anemia. There are no red blood cells. They are suffering from hypoxia. They cannot breathe because there is no oxygen carrier. They are suffering from infections because there are no white blood cells. They are suffering from bleeding because there are no thrombocytes. They are suffering from fractures. Actually, the first complaint, one of the first complaints, I remember it very well from the clinic, is back pain, backache. They're coming with a backache. And, backache, take some, go to the physiotherapist, take some pain relievers, but they come back, and you do an X-ray. Now we don't do X-ray anymore. We are doing CT scan or MRI, and you find a collapsed, fractured vertebra. Or they break their femur or whatever. Because, again, the tumor presses against the bone and erodes the hard part of the bone. And they die in agony, renal insufficiency, because the high level of the immunoglobulin clogs the tubuli of the kidney and so on and so forth. And now they are living, these patients, some of them are being cured, and some of them are living 10, 12 years with high quality of life. So the landscape changed dramatically. Exactly, I always compare it to malignant melanoma, how the life of patients with aggressive malignant melanoma changed with the development of immune therapy, of CTLA-4 and PD-L1 antibodies, and so on and so forth. So this was one of the biggest revolutions in oncology. Here you can see a non-Hodgkin lymphoma. You can see a chest X-ray. You can see the tumor here in the hilum, in the gate of the right lung. You can see here the big arteries. You can see the vertebra, the spinal canal, and you see how the tumor shrank from this to this. So it's typically lymphomas and tumors of the lymph system that is affected by the proteasome inhibitor. Now, what is the mechanism? The mechanism is very important because it's not a chemotherapy. You can add chemotherapy on top of it. The mechanism of action is induction of apoptosis. How? Because all these lymph cells are secreting antibodies. So it's a big factory to secrete antibodies. Now, again, the ubiquitin system, remember, one of the roles of the ubiquitin system is quality control. It should catch abnormal molecules and degrade them. Now, if you have a factory that makes million molecules a minute or 10 million molecules a minute, some of the molecules are defective. Think about a car production line in Detroit, in Ford factory. They make 1,000 cars a day, 2 cars are defective. But if they make a million cars a day, then 200 cars will be defective. So the ubiquitin system can take those cars out. It should take these cars out of the production line, and they will not send them to the franchise to be sold. So that's what happens here. The ubiquitin system is normal. It takes care of but once I inhibit the ubiquitin system, I don't inhibit it completely, but I reduce the level, so these abnormal proteins are coming up because we cannot degrade them all. If they are coming up, they will induce unfolded protein response, the UPR, which is a known response of the cell once abnormal proteins are accumulated, and the UPR will induce apoptosis. So I can kill cell, I can induce apoptosis by a non-chemotherapeutic mechanism, just by leading to accumulation of abnormal antibodies. So I can add on top of it, I can still add chemotherapy, which is a huge advantage in oncology, because we are always using a combination. But 2 chemotherapies are, first of all, are toxic, and many of them are working in similar mechanisms, so they will not be synergistic. In this case, I can add any drug, and the drug will be synergistic to the activity of the proteasome inhibitor. So this is one. I will now go, because we are approaching the time, I will go now to the real development that happened in the last few years, and it's a very important development, and that's molecular glues, or PROTACs. So this is a very important development. Now, what is a PROTAC? A PROTAC is a small molecule that has 2 heads. It has 2 heads. It's a matchmaker. It's a glue. It's again a molecular glue. One head, the authors here really magnified the molecule disproportionately just in order to give you an idea. But it's a small molecule in the molecular weight range of 500 to 1,000. They are all cell-penetrable. So one head, the yellow head, will bind to an E3, to a ubiquitin ligase. Remember? The other head, the red head, will bind to the target substrate. And they will bring them to the appropriate vicinity, so the ligase will ubiquitinate the substrate, and it will be degraded. Now, if you have what happens in cancer and in neurodegenerative diseases, that they are drivers of the diseases. Let's talk about cancer. In cancer, always the driver of the disease will be an oncogene, an oncoprotein. Let it be MYC, let it be mutated p53, let it be RAS, let it be JUN, let it be NF-κB, let it be anything. A protein that should be recognized by the ubiquitin system because it's an oncogene. It's a mutated or pathologic protein that should be normally degraded, but is not degraded somehow. So what do? I force its degradation by matching it, by letting it marry the ubiquitin system, bringing it to the appropriate vicinity to a ubiquitin ligase that otherwise will never recognize it. This ubiquitin ligase is foreign to this green protein. But if I bring them together, it will degrade it. It will ubiquitinate it. It will attach ubiquitin to it and degrade it. So basically, I'm using molecules that are molecular glues in order to force degradation. Now, for those of you who are coming from medicine, if you are reading a textbook of pharmacology, it's mostly antagonist. It's not agonist. You know, in order to reduce blood pressure, we are using beta blockers. In order to curb cancer, we are using kinase inhibitors. So we are always, you know, I would say that 80% of any textbook of pharmacology, of all the drugs that you are buying in the apotheca, are inhibitors. In this case, I have an agonist. I can force it. This is by itself a new concept to pharmacology, not a new, but rather a rare concept too. And it opens the road very wide for the future. Think about RAS. Now we can degrade RAS. They are already degrader of RAS. RAS was one of the most undruggable oncogenes. You know, in ovarian cancer, in pancreatic cancer, in brain cancer, in all these aggressive cancer that have no solution. Now we can start thinking of it all about a solution by using products. So all what you have to do in order to develop a product, well, I oversimplify it, is to screen a library of chemicals. And there are numerous libraries for a high affinity binder to this guy, and you identify the red head. Now, this ligase is kind of any ligase will do it. So all what you have to do is use the yellow one that is already available, and you have to combine the red one that you found in your screen to the yellow one, and you have, in principle, you have now a product. So think about prostate cancer, one of the most common cancers in men. You know, the initial phase is always hormone dependent. It's testosterone dependent. So what you want to do is to degrade the testosterone receptor, right? So now there are degraders. They are called targeted degraders for the testosterone receptor. And there is a big company in the United States, Arvinas. There are numerous companies now that are developing different products for different purposes. Now, the products came from an unbelievable discovery. You know, it's always... I was today in a very interesting exhibit in Athens about Asia Minor in the Benaki Museum about what happened to the Greeks during 100 years ago in 1921. And when I walked out, there was a book that to sign, you know. So I said that people that don't have a past don't have future. And this is, you know, knowing here the history was very important. The products were developed from thalidomide. Now, what is thalidomide? Thalidomide was, only old people like me can remember what thalidomide was. I was already a medical student. Thalidomide was a drug that was given to one of the most common diseases. And the disease is, it's not really a disease, it's a disease of happiness. It's morning nausea of pregnant women. And the United States, the FDA, did not allow thalidomide to go into the United States. They felt that something is wrong with the clinical trials. But in Europe, it was used heavily. And within no time, after it showed up in the market, deformed babies were born that lack limbs, mostly limbs, upper limbs, lower limb, syndactyly, no fusion of fingers and so on. It was the biggest disaster in the pharma industry. The connection was made very quickly because all the mothers took thalidomide during the critical phase, the early phase of their pregnancy, and it was stopped and buried in the ground, never to be heard of. But then the NIH, about 15 years ago, only 15 years ago, in 2005, pulled out many drugs for a program called repurposing, because drugs were developed for one purpose, but then they said maybe it's good for other purpose. And they screened many drugs for other diseases model, And they found that thalidomide can kill any lymphocyte that, if a lymphocyte smells thalidomide, it's dead. And I'll explain to you in a minute why. And thalidomide was very quickly developed as a drug for multiple myeloma. Multiple myeloma is typically a disease of more advanced age, so there was no issue of pregnancy, but nevertheless, the FDA was very careful about it. People thought that if a woman walks in the garden and the neighbor is using thalidomide, she might smell it or whatever. So every patient initially in the United States, the physician had to get a special license, and the patient has to be in a hospital isolated and so on. But then, the fears kind of dissipated, and thalidomide made it into the market. Now, what is thalidomide? You see, it has two heads, one head and two heads. And what it binds Aiolos and Ikaros. Now, Aiolos and Ikaros are the transcription factors that are driving multiple myeloma. Once it binds them, it links them to cereblon, which is the ubiquitin ligase, and they are ubiquitinated and degraded. So there is no transcription factors, Aiolos and Ikaros, that will drive the multiple myeloma, and the multiple myeloma cells are dying because nothing drives their division. On top of it, it was discovered that it also drives another protein, which is called MACE2. Now, MACE2 is a growth factor that is responsible for the development of the limbs. So thalidomide can drive for degradation bad proteins, but it can drive also a good protein that is responsible for embryonic development. And this is the source of the disaster of thalidomide. So now we understand, by understanding the mechanism that thalidomide is basically a PROTAC, we understand also the pathogenic issue of it. There was also an issue with an enantiomer, with the D and L isomers, but I don't want to go into it. But the basic mechanism is that it drives for degradation also a critically important developmental factor that is responsible for the development of limbs in the developing embryo. And now we have not only thalidomide is out now, we have lenalidomide and pomalidomide, and so on. You see here, lenalidomide causes selective degradation of zinc finger proteins, which are also drivers of multiple myeloma in multiple myeloma cells, and so on and so forth. And now the literature is full of them, and it's a big industry now, using the Ubiquitin system as a platform for development of PROTACs. So the PROTACs is basically an unlimited platform for agonists. You can do it for any disease at your likening, and it looks simple. It's not that simple, but nevertheless, at least we have kind of a road. So let me summarize what I told you. We started, as I said, with curiosity. Though we are both physicians, we couldn't expect the future. Like, you cannot expect the future for any development. We wanted to know how proteins are degraded selectively. We did know that we are looking for something special, something unique. I don't know what's special. Something unique, and we discovered it. We discovered the energy dependence, and we discovered the marking, because the marking is the critical thing for being. So you can imagine that there is a shark floating here in the room, and the shark will identify only people that are bleeding. There is a belief that sharks are biting only, they are attracted to blood. I wouldn't do any experiment swimming next to a shark, even without bleeding, but nevertheless. So you need a specific shark. You need what we call a cryptic protease. The proteasome is a cryptic protease. It's inactive until it will find a ubiquitinated protein that was first identified by the Ubiquitin system to be ubiquitinated. So there is a two-step of recognition in order to enter the shredder. So that's what we were looking for, and we didn't know what mechanism we are expecting, but we knew what we are looking for in principle. And this, I think, the guiding line was very important, because otherwise we would have been lost. Because we discovered on the way we discovered catepsins on the way. We were almost trapped with red herrings along the way. But because we knew what we are looking for, we excluded the red herrings on the way, because we knew that they will not answer the question that we had. And I think that I was lucky in the sense to go back to my stupid, naive dream about understanding diseases. Because again, most people don't enjoy it, and not because they are stupid, because, one of the factors that you need in order to be a successful scientist is luck. I mean, and luck, you need the stars to turn in the right direction, and we have no control on that. So, so now I'm finding myself in a position that I can really look from 30,000 feet and see how the health and the impact and the health of millions of people are being affected by our discovery, though we are not developing a drug. Now I'm developing a drug we have a very successful drug, we hope. We are very close to clinical trials now. But all the stories that I tell you about thalidomide and proteasome inhibitors are not ours. So other companies did it, and we are happy with it. Of course, we are happy with it, so we can see it. We can see the impact. Another issue here is very important for governments. And governments, you know, they always come and ask you what you are doing. And you tell them, I'm studying protein degradation. They said, why? You know, it's the taxpayers' money. Why you are wasting it? It's basic research. And basic research is critically fundamental for discoveries. Think about it, you know, they always tell you, do translational research, do something practical, study cancer, bring drugs for us. You cannot discover drugs to cancer without discovering the proteasome and without understanding E3s and the ubiquitin ligases. You cannot do it. It must be, and even, you know, I try to explain to politicians, you know, they say, do applied science. I said, you know, you said it yourself. The word translation means you translate from one language to another. You translate basic research into practical research, into marketable product. Apply, yes. You apply basic knowledge into practical knowledge. So in the word translational and in the word applied, you already embed basic research. Without basic research, there will be no development of science. And there will be no development of drugs, and there will be no development at all. Let it be in engineering, let it be anywhere. You cannot start from the top. Buildings are being built from the foundation. And the foundation is basic research. And this is a very important lesson that politicians, it's hard for them to, because even me, people are asking me, are you studying cancer? You have drugs. I said, yeah, because I'm already 40 years into the business. So I'm in the stage of developing drugs for a work that I did for 30 years without knowing what is a drug. So this is the fundamental issue. And I think that this study represents and epitomizes, not more than any other study, but think about COVID-19. The central dogma of biology has been there for 60 years, since the discovery of the double helix. And people knew that something is coming. The human genome came, understanding mutations, sequencing, but nothing really. And then came COVID-19 and said, oh, look how quickly we developed the messenger RNA vaccine. No, it was not quickly. It took 60 years because the central dogma of biology has been there, not being used, but all of a sudden there was a need and messenger RNA was synthesized and injected and it's working. And now it will be like a storm. Because now now that you know that the principle is working, it will be for cancer, it will be for any disease. You can now develop vaccines and you can change them. The virus changes, yes, the next day you have a vaccine because you make a bivalent one. You don't have to purify a protein, which takes forever. You don't have to attenuate the virus to get a weak virus. You can just go to your keyboard and instruct the machine to synthesize the new message with a mutation in the tail, in the spike protein of the, let it be Omicron, let it be BA.5 or whatever. So think what the central dogma that has been lying there for 70 or 60 years like a dead body, all of a sudden woke up to life and to important life, because the fact that we can sit here today and listen to one another without mask, that's it. This is pure science. People, you know, you just have to explain it to people so they know where they are living. Thank you very much.
Have a seat and I'll go. So, I hope that you have enjoyed all his excellent lecture, especially for the young students. There were also a few important people who came to attend the lecture, which I didn't pay attention at the beginning. And one of them is a major scientist who was also a politician, not a politician, but he did politics, and this is Professor Frankoulis, who has been also a former General Secretary of Research and Technology, and also an active member of the FEPS Council. So perhaps, if I do not see anybody, please excuse me. but it's going to be time just to go through. So now I would like to give to both Aaron and to Rebecca just a small gift about our presence. So thank you both for coming. And please come and receive something just to remember this occasion. And afterwards, I would like both of you just to sign the Librador. Okay, as you did in Benaki Museum, now you have to do it for the Hellenic Pasteur Institute and write whatever you like, so the next generation you will remember your presence. So please, both of you come. So, the lady first.
Thank you very much, Tapis. Thank you very much.
And that's going to be a reception, so Aaron and Rebecca will be with us. So if you like to ask them whatever.